This is really transformative technology.
In structural biology, resolution is everything. The field has really evolved from resolving blobs or general outlines of proteins to now even seeing the details of proteins at atomic resolution. I would compare the advances in CryoEM over the past 10-15 years to be very similar to what the James Webb Space Telescope has opened up in the astronomy field.
I think for all practical purposes, it would have been impossible to do this work without the sort of technology that's been enabled by the Beckman Foundation.
The generous funding from the Beckman Foundation has enabled us to actually establish our own Beckman Center for CryoEM that houses a powerful Titan cryos. We are a vibrant structural community here at the University of Utah. And labs from the campus and from elsewhere come here to use our cutting-edge instrumentation, the Titan cryos, to uncover the principles of cell biology. The microscope enables us to gain information about these little systems and molecular machines at an unprecedented speed. In the short time we've had the machine installed, we were already able to publish 20 peer-reviewed papers, many of which were high impact and shifted dogmas and paradigms in fields.
Using electron microscope here at Utah, we were able to see clearly how a common diuretic drug acts on the kidney transporter protein. Diuretics are cornerstone of treatment of high blood pressure and also fluid retention in the body. Loop diuretics drugs prevent kidney membrane protein from absorbing too much salt. These drugs have been used in clinic for more than 5 decades. But how the drugs exactly work on this, acts on this protein, we don't have a very clear idea about that. So that's what we wanted to understand in the lab. The protein we are studying in the lab they change shapes all the time, so which is very difficult to study using conventional technique. So we have to rely on CryoEM to determine their structures, to see how the protein interact with drugs used in clinic. So based on the structure, we can try to design new drugs that can act on the protein, but can be more effective and has fewer side effects.
In science, these eureka moments are few and far between, and so we're sure to celebrate them when they come. And I would say that our determination of the 3D structure of this unfolding complex was one such eureka moment. Using CryoEM, we've determined 3D structures of an enzyme complex that's directly responsible for unfolding proteins. And what we've learned from our structures is that if you think of proteins as a ball of yarn, these proteins actually need to be unfolded into a state where cells can prepare them for degradation. And this enzyme that we've characterized turns out to be directly responsible for unfolding these balls of yarn into a linear state. And in their linear state, they can be degraded more easily by the cell. This is an enzyme complex that has been known for more than 20 years, long known to be associated with neurodegenerative disease. And for a long time, despite our knowledge of the existence of this enzyme, nothing was known about how it works. And we hope that eventually our structures will be used by others to guide their efforts in designing specific drugs that can dock or fit specifically into our CryoEM reconstructions to guide their efforts in developing therapeutics.
Oh, it's an incredibly exciting time when you see a new structure for the first time. This project starts from some really remarkable, astonishing biology, predatory marine snails. They secrete a venom into the water that causes the fish's blood sugar to crash and therefore becomes so woozy it's unable to swim away from the snail, which can then slither up and just eat it. The insulin is very fast acting, and what our collaborators realized is that this has a real therapeutic potential because speed of action is very important in treatment of diabetes, including in diabetic pumps. So what some of our collaborators did is they took this venom insulin as a starting point and designed a humanized version of it, which should be able to be functional in humans and act very, very quickly. The contribution that we made was to determine the structure of this humanized insulin in complex with the insulin receptor. And by doing that, we were able to learn how it manages to function and maintain its fast-acting capability. And we also learned how to improve it potentially in order to have even greater therapeutic possibilities. So I think the technological breakthrough enabled by advances in CryoEM have really been transformative.
In addition to us using this microscope, we've also helped users from all over to obtain structures of their proteins, and we also helped about 9 different companies to study their samples of interest. In the future, we will also move into the field of cryo-electron tomography that actually helps us to study proteins directly in their natural habitats, inside mammalian cells, inside yeasts and tissues. And it is very important to do this because we can uncover how these multi-component molecular machines actually work and how the wheels are turning to make biology work. Thanks to the Beckman Foundation's generous funding, this has enabled us to establish the Beckman Center for Cryo-Electron Microscopy, and it's continuing to enable us to stay at the leading edge of science so that we can observe biology in action.